Covalent modification which occurs in post-translational modification. Example, phosphorylation. As name suggests, the addition of phosphate group makes changes in protein. The hydroxyl group of certain serine, threonine, tyrosine residues of some proteins are enzymatically phosphorylated by ATP and phosphate group, which add negative charge to these polypeptides. And the functional significance of these modifications varies from one protein to the next protein. Example, as you can see, the phosphorylation of casein is an important post-translational modification occurring after the synthesis of polypeptide chain in the Golgi apparatus of mammary epithelial cells under the action of protein kinase, and which mainly has many phosphoserine groups that bind to calcium, and then calcium phosphate and amino acid 3 uh, are valuable to suckling young, so casein efficiently provide these essential nutrients to these youngs. Babies. Carboxylation. Extra carboxyl group may be added to glutamic acid residues of some proteins. Example, mainly involved in blood clotting factor, pyruvate carboxylation cycle. So what happened during blood clotting mechanism? The blood clotting protein mainly contain a number of carboxyglutamate residues in its amino terminal region introduced by enzyme that required vitamin K. These carboxyl group bind calcium, which is required to initiate the clotting mechanism. Glycosylation, as a name suggests, addition of a glucose molecule is known as glycosylation. So, addition of carbohydrate molecule to the polypeptide chain and modifying it into a glycoprotein. Many of the proteins that are destined to become a part of plasma membrane, as you can see easily in plasma membrane, they contain glycoproteins, or to be secreted from the cell, have carbohydrate chain attached to the amide nitrogen of asparagine and linked or hydroxyl group of serine threonine, which is known as O-linked. So N-glycosylation occur in endoplasmic reticulum, while the O-glycosylation occurs in the Golgi complex. Sulfonation. So sulfate modification takes place by the addition of sulfate molecule, and these modification of protein occurs at tyrosine residues. Methylation. The transfer of one carbon methyl group to nitrogen or oxygen to amide side increase the hydrophobicity of protein and can neutralize a negative amino acid charge when bound to carboxyl acid. Methylation is mediated by methyl transferase enzyme and acetylinosyl methionine. It is a primary methyl group donor. So, after protein synthesis, protein synthesis is inhibited by many antibiotics and toxins, as the protein synthesis is central function in cellular physiology and it is primary target of many naturally occurring antibiotics and toxins. So, several antibiotics and their target site are listed in this table. As you can see, there is a list of antibiotics, their target cells, prokaryotes and eukaryotes, their molecular targets like 30S. Some antibiotics attach attacks 30S subunit, some 50S, some EFGU factor, some and another EFTU factor. So all this table represent their molecular target and their consequences. So as example, you can see there is a permomycin, which mainly target cell is prokaryote cell. Adjacent to the adapter or A site codon, anticodon interaction site in the 30th subunit, and consequence it increase error rate during the translation by decreasing selectivity of codon anticodon pairing.